Hi, I'm Anna Warren, and we're from Connecticut College. My group that included Rachel Cantor, Karin Liston, and Lena Odell did a study on the effect of early developmental lead exposure on adolescent spatial learning and memory and aggressive/antisocial behavior. The goals of this study were to expand on previous research done by Connecticut College that measured the effects of developmental lead exposure on spatial learning and memory and was assessed through the Morris Water Maze, the Cincinnati Maze, and the Radial Arm Maze. In our study, we utilized the Barnes Maze to assess the effects of developmental lead exposure on aggressive and antisocial behavior, as well as on working memory. In terms of the societal benefits that experiments like this can have, environmental lead exposure can occur in humans in many forms, such as paint used in houses, toys, soil, and water exposure. Previous research has shown that chronic ingestion of small amounts of lead can cause cumulative neural deficits, such as IQ deficits, learning disabilities, and behavioral problems. Additionally, evidence from animal studies has suggested that exposure to environmental lead during early stages of cognitive development can have detrimental effects on learning, memory, and cognitive performance. It's important that we develop sensitive behavioral methods and useful animal models to evaluate the effects of environmental toxins and pharmacologic agents on learning, memory, and social behavior. This study was done that focused on the comparison between the increase in amount of atmospheric lead in the 1950s and subsequent decrease in the 1980s to the similar rise and fall of aggregate assault rates between the 1980s and 2010s. The intent behind this study was to demonstrate that the presence of lead in the atmosphere had a physical effect on the areas of the brain that control aggression.
Hi, I'm Lena Odell, and I'm going to be talking about the methods of our experiment. So for this experiment, we had 2 pregnant Sprague Dawley rats that were housed separately, starting immediately after their arrival at our facility, at which point the rats had been pregnant for about 10 days. The first was given water, which had 0.4% lead acetate, and the second was given tap water. Once the dams gave birth, the pups drank the same water that their respective moms was drinking. The pups, after being weaned at 3 weeks, underwent the play fighting behavior assessment, where the litters were observed for 5 minutes and the number of pins that occurred were recorded. Once the pups were 6 weeks old, they underwent the Barnes Maze testing over a 10-day period. The first day was habituation, in which the animals were adjusted to the space without being recorded, and it then followed 5 days of acquisition, where their performance on the Barnes Maze was recorded, followed by 1 day of acquisition probe testing on the Barnes Maze, where the behavior was assessed again. The rats then went through the aggressive behavior assessment, where based on the resident intruder paradigm, the rats were tested for offensive aggression against a stranger or a rat from the other litter. So this is the Barnes Maze. We utilize the Barnes Maze in order to measure the effects of developmental lead exposure on spatial memory and learning. Previous studies have used the Morris Water Maze, the Cincinnati Maze, and the Radial Arm Maze in order to measure this. These other methods cause higher levels of stress to the animal via forced swimming and food deprivation. One of the big benefits of the Barnes Maze is that it's relatively low stress, and it allows for measurement of spatial reference memory, spatial working memory, and cognitive flexibility. There are also no previously published studies which use the Barnes Maze to measure lead exposure on spatial memory. So the setup of the Barnes Maze, it's brightly lit from above, and it includes 1 escape box, which you can see at the top left-hand corner. They also have false escape boxes, which are under all of the remaining holes. These have patterned maze cues, which are placed on the surrounding walls, and they're visible to the rat, which can then be used as a visual reference to learn the location of the escape box. So this is the aggressive behavior assessment. So it's based on the resident intruder paradigm, which has been used to measure aggression in rodents. The rats were paired with a rodent from the opposite litter in a clean cage, and each pair was observed for both aggressive and non-aggressive behaviors over a period of 10 minutes. Aggressive behaviors included rearing, lateral threat, upright posture, clinch attack, keep down, and chase. Non-aggressive behaviors included move towards, social exploration, and anogenital sniffing. So these 2 images are examples of aggressive behavior. The first is the clinch attack, in which one rat attacks another and they end up scuffling. And the lateral threat is when one uses their side in an attempt to interrupt the other rat's social interaction and attack that way.
Hi, my name is Conn, and we are going to learn about the results section. We found that lead-exposed rats in both group housing and isolated housing made more working memory errors than those in group housing. Lead made more preservative errors, and isolated rats made more primary reference errors. This tells us that lead exposure plays a role in learning acquisition and decreases learning ability. We also found that there was no correlation between escape latency and lead exposure. There was no statistical significance between the group water and the lead rats among isolated water and lead rats. This shows that lead may have an effect on learning acquisition, but not necessarily on memory. Here we found that overall lead-exposed rats displayed more aggressive behavior. We also found that there was more non-aggressive behavior in isolated rats than in group water rats. We also found that it is statistically significant that both isolated rats and group lead rats displayed more aggressive behavior than their water counterparts. To conclude, our study shows that lead-exposed rats have a more learning acquisition errors in total. Like we've said in previous slides, lead may have an effect on learning acquisition, but not necessarily on memory. We also saw in our research that isolated rats and group rats had more aggressive behavior than water rats. This confirms previous research that lead exposure causes more aggressive, violent behavior in humans. For future directions, we propose that there should be a different way to assess learning memory to confirm that lead does not have an effect on learning memory. Thank you for joining us as we talk about our study, and we'll now open the floor to questions.